Hi students, today's topic is euchromatin and heterochromatin. You are all clear with what this chromatin is and the detailed structures of nucleosome model and its packaging were discussed in our earlier video. So now today we will be seeing different types of chromatin. This chromatin can be of two types, either euchromatin or heterochromatin, that is the chromosome or the chromatin exists in two morphological types, either it is a euchromatin form or it is a heterochromatin form. Now what is euchromatin? Euchromatin are the regions of chromatin which are less condensed or less tightly packed, and they will be transcriptionally active regions, while heterochromatin are the regions which are so tightly packed or so compacted, and because of that they will be transcriptionally inactive regions. So since these heterochromatins are transcriptionally inactive or tightly packaged regions of the chromosome, they can be of two types again, either constitutive heterochromatin or facultative heterochromatin. Constitutive heterochromatins are the regions that are always heterochromatic or that are always tightly packed, that will not loosen up for any reason, that are permanently inactive with regard to transcription. While facultative heterochromatins are the regions which are tightly packed, but at certain situations or certain needs, when demand occurs, that region will loosen up or that will convert to euchromatin. So such type of heterochromatic regions which is reversible or which can convert to euchromatin depending upon the needs, that region is called facultative heterochromatic. So these are the points related to heterochromatin. Heterochromatic regions are the relatively condensed or tightly packed regions of the chromosomes, and since they are tightly packed regions, they will take up the stain and hence will appear as darkly stained regions of the DNA. And it is of two types, constitutive and facultative. All these details have been discussed. And regarding the euchromatic regions, these are the less compactly packed regions and they will be less deeply stained regions. They are transcriptionally active regions, and most of the regions of the chromosome will be euchromatic. You must take into the point that the main point is the genes which are actively transcribing, or the regions of the chromosome which are more densely having the genes, that regions or the regions which are having more dense number of genes will be euchromatic regions, because they need to be transcribed, that the genes need to be expressed. For expression, this DNA or the DNA has to be exposed, that is, it must uncoil for the transcription process to occur. So where the genes are mostly situated, that regions of the chromosome will be euchromatin, while those other regions which are not having so much of genes, that regions will be heterochromatic. We will see the details. So look at this figure. You are familiar with this, I'm sure, and here you can see the packaging details of chromosome. With this we have gone through in our previous class. This DNA, it will undergo packaging. This is the nucleosome model, and again it is condensed to form the solenoid, and again scaffolding occurs, and finally it forms the chromosome like structure during the cell division process. And for a gene to get expressed, is it possible if it occurs like this, in this state, is it possible that the genes to get expressed? No. for the gene to be expressed, expression means the DNA has to undergo transcription to form mRNA, and that mRNA has to undergo translation to get converted into the proteins. So for transcription to occur, the enzyme which is responsible for transcription is the RNA polymerase enzyme. So if the RNA polymerase has to acquaint with the DNA for the generation of mRNA, it has to scan through the DNA. So obviously the DNA has to get exposed. So this tightly packed, this state, is it possible for the RNA polymerase to scan through this DNA? No, right? So for this, for the gene to get expressed, or for the transcription process, it has to loosen up, or it has to revert back to these less tightened or less packaged structures. That is the mostly it occurs in the chromatin structure or the nucleosome model. So that type of chromatins, we call it as euchromatin. And if the region in the chromosome, there are certain regions which are always heterochromatic and those which are euchromatic regions. The euchromatic regions, say for example these regions are having genes which are always necessary, or which are always needed to get expressed in an organism. So that regions will be in euchromatic form because it needs to be get expressed once and every while. So for every one and while, it has to loosen up and again get back to the condensed form. So for that reason it will be occurring as euchromatic regions, while those other regions, like for example this centromeric region and these telomeric regions, there the chromosome is not having so much of genes which are always needed. So that regions, these are the gene scarce region. So here the gene density is so low in the centromeric region, and then the telomeric region the gene density is so low. So that regions will be having only repetitive DNA sequences, or we call it as junk DNAs. That repetitive sequences are there which are meant as raw materials for future evolution process. So there we don't have any genes. So that region we don't need it to be in the form of a euchromatic region. So that regions will be always in the heterochromatic regions, particularly constitutive heterochromatic regions. This is how a heterochromatin and euchromatin occurs. This is, you can understand from this figure also. This is so tightly packed and this is loosely packed. Two types, difference between heterochromatin and euchromatin. See in this figure, the telomeric region and the centromeric region, which is orange colored regions, indicate the heterochromatic regions because these regions are gene scarce region. Gene density is very low in that region. Euchromatic regions are stained purple colored. Their gene density is high, so they are euchromatic regions and purple colored. This is another difference between constitutive heterochromatin and facultative heterochromatin. Example for constitutive heterochromatin is centromeric region and telomeric region. This is a DNA and this is a chromosome, and this telomeric region are the end regions of a chromosome. Central region is the centromeric region. So centromeric region and telomeres are examples of constitutive heterochromatin. They are always these compactly packed regions, while in other regions they are less compactly packed. That is clear from the diagram. So they are euchromatic regions. Facultative heterochromatin's example is bar body. And centromere, what is a centromere? Centromere is the central portion of, not always the central portion, but the region which attaches, which is the center point or an attachment point of two chromatids or the two sister chromatids. and here it is heterochromatic region, constitutive heterochromatic region, densely packed or tightly packed, deeply stained region, and these regions are occupied by repetitive DNA sequences. Gene density is low there, and this is the point which forms the kinetochore. So centromere and kinetochores are the important regions which are responsible for equal segregation during the reproduction, cell division to occur. So certain proteins will come and attach to the centromeric region and form the kinetochore complex, and this kinetochore complex are the point of attachment of spindle fibers which are responsible for the segregation of chromosomes. And the size of the centromeric regions may vary from 200 base pairs to 40 kilobase. So that is all about euchromatin and heterochromatin. Hope you all understood. Thank you.